dive straight in. Biology higher, paper 1, make sure you got the right paper. Question number 1 was just a connect the lines. Nucleus, eukaryotic cell, permanent vacuole, plant cells only, plasmid, which is a ring of DNA, is found in prokaryotic cells. You get 2 marks for all 3, you get 1 mark if you have at least 1 correct. So you have none right, you get no marks. Question 2. Try and take the time to see if you could have labelled it first and then check it against which box is correct. Vacuole ribosome cell wall, the only row which fits that is that bottom row at the bottom. And then question 1 continued. We have 2 microscope slides. The first one is out of focus. The second one, they are very far away, so I can't actually distinguish them easily. How would the student adjust a microscope to see slide A better? They have to focus it. You must mention the word focus. It can be as detailed as turn the fine focus until the cells are in focus, or you can literally just say focus it, but you need to have the word focused somewhere. If you see underlined anywhere in my answers, that means you have to have that word. For slide B, you are too far out, so you need to zoom in effectively. It is worth 2 marks. Make sure you note every time you do a question how many marks it is out of. So there's 1 mark for saying you are adjusting the objective lenses, why or how, to a higher power to increase the magnification. So 1 mark for objective lens, 2nd mark for increasing magnification. Next question is, now that you have the cell in image, this is how long that image is, 112 millimeters. That's your image length. The real length was 280 micrometers. What is the magnification? I remember it by saying I am image over actual and magnification. The question was asking for magnification, so I cover it. The answer is going to be my image length over my actual length. They need to be in the same scale though. So the first mark is for converting. Either convert the millimeters into micrometers or the micrometers into millimeters. But when you do your calculation, they must be in the same scale. So both of mine are millimeters. They both need to be millimeters. 1 mark for the conversion, 1 mark for showing what you are doing. So this calculation gets you a mark, and then 1 mark for the answer at the end, 400. Give yourself a total out of 9. Make sure you're being harsh now so you get better later. Question number 2, coronary heart disease. What is a non-communicable disease? Well, it's something which is not caused by a pathogen, so it cannot be passed on. Again, make sure you look at how many marks things are worth. Explain how coronary heart disease can cause a heart attack. A heart attack is when your heart muscle dies. So why would the heart muscle die? From a blocked artery. Well, there's less blood getting to it, so it gets less oxygen, so the cells can't respire. There's a little bit of a sort of story going on. Reduced blood flow, so less oxygen, so the cells can't respire. 1 mark, 2 marks, 3 marks. 6 marker, your first 6 marker. Explain how lifestyle and medical risk factors increase your chance of getting coronary heart disease. Explain is a why question. It is mostly just knowledge. So pause here and have a look at the mark scheme. But my main point for level 3 response, where you're getting your 5 or 6 marks, there had to be a link. So smoking causes high blood pressure. Obesity causes or is a risk factor for diabetes. So linking the lifestyle and the medical risk. Moving on to question 3. Have I got 3.1? Where have you gone? Oh, it's on the side of this page. Question 3, the first 2 straightforward tick boxes. What's the correct balance equation for photosynthesis? Well, the word equation, carbon dioxide, water, glucose turns into glucose and oxygen with light. The only option which fits is this box down here. This one is our molecular formula of glucose. What type of reaction is photosynthesis? Aerobic means with oxygen, and that's to do with respiration. Endodermic takes in energy. We need light for photosynthesis, so it's endothermic. Exothermic releases energy. Oxidation is a loss of electrons. Then on to the practical associated with photosynthesis. We have LED light, which is quite cold, doesn't produce much heat. pondweed and sodium hydrogen carbonate, which is a source of carbon dioxide, so that is also not a limiting factor. Suggest 2 ways it could be improved to measure the rate of photosynthesis more accurately. There are 4 options here. 1, instead of just measuring bubbles, they are quite inaccurate, unprecise. You will not be getting a true value of your rate. You can measure the volume of gas that's being given off, capture it in a measuring cylinder or gas syringe. 2, you can measure for more than 1 minute and then divide by the amount of time you measured for. Number 3, take a video and then recount your bubbles. And 4, repeat at a distance and calculate a mean. You must mention calculating a mean if you say you are repeating. You do not get the mark if you just say repeat at each distance. You must tell me why it makes it more accurate. LED source doesn't get hot. Why is it important it stays at constant temperature? Because temperature affects rate. You need to have the word rate of photosynthesis or bubble production instead of the word photosynthesis. Why does it do that? Explain why. Because reaction is enzyme controlled and enzymes work faster when they are at a higher temperature. Light intensity can be calculated by the inverse square law. What that means is if you get twice as far away, you actually get 1/2 squared, so a quarter as much light as you did at that distance. It's asking you to prove it, and there must be some sort of evidence of calculations. So the way I have laid it out is I have a distance which is doubled. So how does it change when it is doubled? So I'm using the question to help me answer it. As it is doubled, what happens to 1/d squared? So 5 squared, 1/25, 10 squared, 100, 1/100. That is the same as it being quartered. So as distance doubled, light intensity quartered. The way it breaks down to show you the mark scheme, making sure you have evidence of 2 distances that have doubled. 5 to 10, 25 to, that's your distance doubled. Or you could have used 10 to 20 because that's also a doubling. Calculate the 1/d squared for those 2 distances, as I have here. And then a final sentence, as distance doubles, light intensity is quartered. What happens to your results as you go along? It's asking you to estimate. If you go to 40 centimeters, what happens to your light intensity? Well, that is a doubling of 20. So it's going to be a quartering of the number of bubbles at that time. So 2 bubbles is my estimate. Why does that happen? Give a reason. 2 marks, remember. There is very little light for photosynthesis. Once it is underlined, you must use that word in your answer. If you haven't written it, you do not get the answer. You can change this question or this investigation to investigate the rate of carbon dioxide concentration. How would you do that? And then what else would you need to control? The first step is, what is your independent variable? Carbon dioxide concentration. How do you change that? Sodium hydrogen carbonate is your source of carbon dioxide in this experiment. So to change your independent variable, use different concentrations of sodium hydrogen carbonate. Use these bullet points as a mark scheme as you write your answer. They have told you what they're looking for. How would you change it? Use different concentrations. And then 2 controls. There are 4 ones you can pick from. Your distance from the lye source must be controlled. Temperature of solution must be controlled. Use the same plant in each experiment and give the plant time to equilibrate to make sure you are actually measuring the photosynthesis rate for that concentration of solution. If you start measuring straight away, it might not have got used to or be responding to the solution it is in. So you need to give the time for it to equilibrate. That is question 3. Question number 4 is another practical. It is the osmosis practical, which you have done with potatoes or seen with potatoes. This time it is a carrot, but the practical is exactly the same. If you are more concentrated than the solution that surrounds you, water will move into your cells. So the most extreme version of this is pure water. Water will move into the carrot because the carrot has more sucrose in this case inside it than outside. Other end of the spectrum, the outside is more concentrated, the inside of the carrot is slightly more dilute, so water leaves your cell and you end up losing water. That's going to be this extreme. Suggest why the student calculated percentage change rather than directly the absolute mass change to control for a starting mass being different. It means you can use the same size and shape of carrot, but you don't need to make them exactly the same mass. You are still being able to change. So you perceive a difference when you do percentage change. There is a percentage change question later, so I won't show you how to do it now. Question 4.2 is draw a graph. Use these bullet points as your mark scheme. Suitable scale and label for your y-axes. Suitable scale, 5 on each side. If you do not have the label, you do not get that mark. 1 mark for the correct scale and label. Plotting the results, here are mine. I've done some annotation previously. They are worth 2 marks and you must be within half a box. Having a plotted point there would be incorrect because you are a whole box away. You must be half a box. Take care and time in plotting your points. 2 marks for all 5 of them being correct. It will be 1 mark if you get 4 or 3 correct and no marks if you get 3 or less of those points correct. Then finally, a line of best fit. Mine's not entirely perfect, but I've got some below the line, some above the line. So on average, my line goes through them. And 1 mark for that line of best fit. The next question, estimate the concentration of sugar inside the carrot cells. You have to use your own data, so do not use my number. The way I got my number was, where does the mass not change? And it's exactly where it crosses the x-axes. Before this moment, the cells gained water, so got heavier. After this moment, the cells lost water, so got lighter. At exactly this stage, there was the same amount of movement into the cell as there was out of the cell. So that changed, there was no overall change in mass. The same amount of water moved in as left. That means that the concentration of the cell was the same in as it was outside. Read your graph carefully. Do not round. Mine is exactly 0.44, so that is why I've put 0.44. Explain why the mass of the carrot in 0.6 molar decimeter per decimeter cubed sugar solution changed. I don't actually know which way it's changed, so mark number 1 is for stating the obvious. The mass has decreased. What happened to the mass? 0.61, the mass has decreased. Why? Because water has been lost through osmosis. You have to have loss of water and osmosis in your answer. They are underlined. You must talk about both those things. Through a partially permeable membrane, the second half of your definition of osmosis. Why? The explanation. I've written 2 different versions of this. One is as the sugar solution outside the carrot was more concentrated than the cells. Probably sugar solution, just to be thorough. Or a different wording, the carrot cells were more dilute than the outside solution. And osmosis is movement of water from a dilute solution to a more concentrated solution. The student repeated the investigation with some boiled carrots. Why did it not change mass? The membrane was damaged, so I'm missing this key part of osmosis. The membrane was damaged, so I'm not going to be able to see osmosis because I haven't got a partially permeable membrane. Question number 5. Let's just make sure I can show you. Question number 5 is about measles. Suggest one reason why the actual number of cases in England and Wales might be higher. So these are the number of people reporting that they have measles. Maybe people don't go to the doctor. The doctor might not send their sample for analysis. And some cases might not be confirmed, so they won't be added into this amount. That all gives you an underestimate compared to the true number. Calculate the percentage decrease. Any sort of percentage change question is, what is the difference over the starting value? So it's asking from 2012 to 2015, what's the difference? 2030 minus 91 over the starting value. So this is the calculation, put it through my calculator, 95.5% decrease in the number of cases from 2012 to 2015. Why might there be a decrease? Well, vaccination. Explain why vaccination works. So most people are immune, so do not become ill. Immunity means you don't get ill in the first place. You need to say those two things explicitly to get the mark. Therefore, there is less chance of unvaccinated, remember not everyone is vaccinated, unvaccinated individuals being exposed to the virus because there aren't as many ill people around. So that is why you have to explain why it reduces the spread of measles. Figure 8, from question 5.4, we have a antibody graph. How much does your antibody level change the first time you're exposed to a pathogen compared to the second time you're exposed to it, whether it is by a vaccine, in which case it is safe and you are being prevented from getting ill because this is a safe way of getting that recognition of a pathogen. Question slide 5.5. Explain that graph and how there is a difference in the antibody production here versus the antibody production here. There are two main parts to this answer. What are the differences in the graph between here and here? And then explaining those differences. Pause, have a read, mark your answer appropriately. If you would like to see the mark scheme version, I will put that up now. There is the mark scheme version. It's basically exactly the same. Question number 6, and the final question of the paper, is a question about the stem cells. Where do you find them in a plant? You find them at the tip of the root and the tip of the shoot. If you don't put the word tip, you do not get the answer or the mark. Or there is a place called the meristem. That is where you find stem cells in the actual stem of the plant. But root tip and shoot tip, that is where it grows from. Economic use of plant cells. You can make clones and you can make loads and loads of the same plant. Maybe you have a really great cabbage plant that you love, makes great cabbages. Now you can make loads of that cabbage. Embryonic stem cells divide by mitosis. So that is where your cells double and your daughter cells are genetically identical. Here is a representation of that. Mass of DNA at the start was 6 pictograms. A pictogram is 1,000th of a nanogram. So this means 1,000th. So on our normal scale, we tend to go down to nano. You've got grams, then milligrams, then micrograms, nanograms. A pictogram is another 1,000th. How many grams is that? Well, it's 10 to the minus 12 grams. Question 6.4. The overall time taken for that to be complete was 15 hours. How many hours does the cell actually spend in mitosis? So 28 degrees of 360 is in mitosis. So that is my fraction that is in mitosis. Times it by 15 hours. And then 3 significant figures. If you have not done it to 3 significant figures, you lose a mark for not reading the question correctly. So be careful about reading the question correctly. 1 mark for the correct answer, 1 mark for rounding it correctly. Describe what happens in these 3 stages. We've got stage 1, stage 2, stage 3. In stage 1, you get 2 marks, 1 for saying the cells grow, 1 for saying the DNA doubles. You need to mention DNA in stage 1. During stage 2, mitosis, the chromosomes are separated and the nucleus divides. So you go from being 1 large cell with lots of DNA to 1 large cell with now 2 distinct nuclei. And then finally, stage number 3 is it splits down the middle and you end up with 2 genetically identical daughter cells to the beginning. There's only 1 mark for that last pet. Final question of the exam, and there are lots of advantages and disadvantages. It looks a bit messy here. I'll give you the full mark scheme. 2 advantages and disadvantages of therapeutic cloning, where you give a patient some cloned, so it is their genetic information, their DNA, but you put it into a stem cell which will divide. All of these stem cells now have the patient's DNA. So why is that advantageous, and it's a potential theoretical use of stem cells? Why is it disadvantageous? Advantages, you can treat some diseases which we couldn't normally cure. These are all the same mark. So if you have multiple of these, you do not get 2 marks, you just get the 1 mark. You can get any cell type. These can turn into liver cells, or heart cells, or muscle cells, or nerve cells. You can get any cell which is needed. They are unlikely to be rejected because they have the same DNA. You can make lots and lots of cells. You can potentially grow a whole new organ. And then some of these could be used for research. And finally, organ transplants require a waiting list. They are not just everywhere, but now you can grow them, you might be able to reduce the waiting time. Disadvantages though, where have you got your egg cell from? That is a potential life. There aren't that many egg cells around. Again, you need a donor to give those egg cells. And donating itself has risk factors. We don't know the side effects of giving these stem cells to patients because it is such a brand new treatment, so there may be some long-term side effects we do not know about. And also it's just a really difficult procedure. It has got a poor success rate. That is Biology Paper 1. Hopefully that run through let you mark. Please make sure you are being harsh in your marking, and also if you need to revisit, go back and revisit and pause as needed.